All right, so again, welcome to the OpenSim webinar. My name is Jennifer Hicks. I'm the OpenSim R&D Manager, and I'll be serving as the moderator for the webinar today. I'm pleased to welcome today's presenters. Massimo Sartori is joining us from the University Medical Center Gottingen in Germany, and Claudio Pizzolato is joining us from Griffith University in Australia. We're coming from all over the world today. So first, let me tell you a little bit about OpenSim. It's a freely available software application for visualizing musculoskeletal structures and simulating movements of humans and animals. The application includes tools for general purpose inverse dynamics, optimization to estimate muscle and joint forces, methods to create simulations for motion capture, and then tools to analyze and visualize the results of your simulations. So the first goal of our webinar series is to showcase cutting-edge research that is being performed with the OpenSim software platform. OpenSim is also a growing and geographically diverse community of users. So the second goal of our webinar series is to provide an easy platform for the OpenSim community to communicate and collaborate. Before we get started, I have a few quick reminders about the format of the webinar. Questions will be addressed at the end of the presentation using the Q&A panel. So that's the first reminder. And then the second, if you need additional technical help, you can consult the guide on our website. So now I'd like to introduce Massimo and Claudio. First, Massimo Sartori. He's a postdoctoral research scientist at the Department of Neurorehabilitation Engineering at the University Medical Center Gottingen in Germany. His research focuses on the development of methods that bridge the gap between neural and functional understanding of human movement. And then he's working to translate these developments to advanced neurorehabilitation technologies. We're also excited to have Massimo visit us here at Stanford for the summer as a visiting scholar in the National Center for Simulation in Rehabilitation Research. In addition to Massimo, Claudio Pizzolato is joining us from Griffith University. Claudio received his bachelor and master's degree in mechatronic engineering at the University of Padua, working with Massimo there as well as Monaco Reggiani. Now he is completing his PhD at the Center for Musculoskeletal Research at Griffith University under the supervision of Professor David Lloyd. His work is focusing on software optimization and real-time application of neuromusculoskeletal models. So we're excited to have them both here with us today to give this webinar on muscle excitation-driven musculoskeletal modeling and future applications to neurorehabilitation technologies. So with that, take it away, Massimo and Claudio.
All right, so thanks, Jen, for the nice introduction. And hello, everyone. It's a real pleasure for me to have the possibility to give an OpenSim webinar today. So with this presentation, I'd like to talk about part of my research on neuromusculoskeletal modeling. And in this, I also would like to give an insight on how some of this application that I will be presenting can be applied to develop effective solutions for neurorehabilitation technologies. Also, towards the end of the presentation, Claudio, who's currently in Australia, will give a demonstrations on how some of the methodologies that I will be presenting in this talk can currently be interfaced with OpenSim. So the problem of understanding human movement is a fundamental question that spans the fields of neurophysiology and biomechanics. And a main limitation here is the current inability of bridging between the neurophysiologic events happening in the humans, such as the firing of spinal motor neurons in the spinal cord and the associated musculoskeletal functions in upper or lower extremity, in which muscles are activated to actuate multiple degrees of freedom simultaneously. So the ability of bridging between these two worlds is important to truly understanding human movement. And the ability of understanding the mechanisms underlying human locomotion is important to develop effective solutions for individuals who are affected by neuromuscular impairments. So a potential solution for bridging between these two worlds is given by neuromusculoskeletal modeling, and this in fact allows converting estimates of neural excitations into predictions of the associated musculoskeletal function. And that's what I'd like to talk about today in this presentation. So the very long term in the title, muscle excitation-driven musculoskeletal modeling, simply means that we will be modeling the dynamics of the muscular and the skeletal system as controlled by the nervous system. So the idea here is to collect experimental data reflecting the dynamic of the neural drive to muscles, and use these information to perform muscle-driven simulations to understand how muscle contract, generate force, and contribute to generate a final movement. And in this context, it is possible to collect experimental data about the dynamic of the muscles at different levels of muscle pathway.
Massimo, sorry, could you increase the volume on your microphone? I'm having a hard time hearing you.
At the maximum level.
Can you move a little bit closer to the microphone?
Yeah.
That's a little bit better.
Yeah, is it better now?
That's a little bit better, yes.
Okay. Let's try to as close as possible.
Thanks.
Okay, so in this context, we can actually record experimental data reflecting the dynamics of the neural drive to muscles at different levels in the neuromuscular pathway. So at the beginning of my presentation, I will first of all talk about how to use experimental recordings of EMG signals to characterize the excitations of muscles and use this as an input for musculoskeletal models. I will then talk about how it is currently possible to reduce the complexity of the EMG signals by exploiting the theory between muscle synergies, and this allows us gaining an insight on how the sensor pattern generating, which we believe to be encoded in the spinal circuitries, act towards the activations of multiple muscles during motor tasks. Finally, I will show how we can use high-density electromyography to gain an insight on how individual motor units into the muscles are activated. And this is a great opportunity to have a direct estimates of the activity of motor neurons in the spinal pathway. And this is very helpful to better understand the mechanisms of muscle excitations and force production. In order for this to happen, it is very important to develop multi-level models that account both for the neurophysiologic level and the musculoskeletal level. And that's important because although we have little knowledge about what happens at one side or what happens at the other side, we still do not have relevant information to associate events at one level to events at the other level. So in this presentation, I will first of all introduce our excitation-driven musculoskeletal model. I will show how we can use estimates of neural excitations recorded at different levels in the neuromuscular pathway and use this to predict the resulting functions in the musculoskeletal system. I will then show how this can be used to implement effective solutions in the field of neurorehabilitation technologies. And then Claudio at the end will show how our model can be used from OpenSim. So I'd like to start off the presentation by explaining how we can use experimental EMG data as an input drive for the model. So EMG data are probably the most viable way to record the activity of a large number of muscles during highly dynamic tasks. And in this context, our model is able to use to reproduce all the transformations that take place from the onset of EMGs until the productions of force into multiple muscles and joint movements into multiple degrees of freedom. And the benefit of using EMG-driven modeling is the possibility of performing muscle-driven simulations without making assumptions on the way muscles activate or share a load across the joint. And that's because experimental EMGs, which actually reflected the actual activations of the muscles during the recorded movement, are used as direct input drive in the model. And this is very important to characterize a number of mechanisms in the muscles that would be difficult to be characterized otherwise, including muscle pre-activations and muscle co-contraction. So as we can see, the model requires input data reflecting the human movement. And in our studies, these data were recorded using a gate laboratory. So we actually use surface electrodes which we placed on the major muscles in the lower extremity, and we placed on the subject retroreflective markers, and we recorded the three-dimensional positions of this marker using multiple cameras surrounding the subject's movement. And then the subject was able to walk on an in-ground force plate that we used to record the foot-ground reaction forces generated during movement. And all this experimental data were then used with OpenSim to produce a number of input data that we needed to calibrate and to validate the model. And we'll explain this later. So with the next slide, I would like to spend a little bit more of time to explain what happens in this step, and then I will talk about how the model works. Before moving to the next slides, though, I'd like to stress the fact that the EMG-driven model that I will be presenting in the next few slides was developed starting from the previous contributions of many people and many institutions. And with this slide, I'd like to thank, if not all of them, at least some of them that I know for sure. So the characterizations of human movement from experimental data was composed of 4 steps in our methodology. So in the first step, we use marker trajectories recorded during functional tasks, and we use this to actually locate the joint centers of rotations at the hip, knee, and the ankle, and we also locate the helical flexion extension axis at the knee. And we do this using solving a numerical optimization problem. In the second step, we take a generic OpenSim musculoskeletal model and we scale it up or down to match the anthropometry of a subject. In the third step, we use marker trajectories recorded during dynamic tasks, and we solve an inverse kinematics problem from which we get estimates of 3-dimensional joint angles. And then we use these estimates together with ground reaction forces to solve an inverse dynamic problem that we can use to actually gain estimates on joint moments. And we do this using dynamic simulations of motions. I actually hope you can see the simulation quite smoothly, despite the limitations in broadcasting of WebEx. So at the end of the simulations, we have experimental joint angles that will be needed later on in the model, and joint moments needed for calibration and validation. So at the end of this step, we have all the input data that we need to run the model. So we have joint angles extracted from marker trajectories, from an inverse kinematic simulations in OpenSim, and we have EMG signals recorded experimentally. And that's what the models look like. So the model is composed of 4 main blocks, as we can see. In the first block, EMG signals are converted into muscle activation. So in this block, we basically account for the electromechanical delay, which is basically the time difference between the onset of the electrical activity in the muscle and the actual time where muscle physically contract and produce force.
And in this block, we also account for the nonlinearity that exists between muscle excitations and muscle force. In the second block, we use estimates of joint angle to compute the kinematics of muscles, including muscle tendon length and movement arms. In the third block, we combine together muscle activation to muscle tendon length, and we generate estimates of the force produced by the muscle. And once we have estimated the force produced by a muscle in a specific instant of time, we can then project this force onto multiple dimensions, onto multiple degrees of freedoms. And we do this by combining the force with the respective movement arms. So in this way, we can understand how a single force produced by a single muscle can contribute to the simultaneous activations of multiple degrees of freedom in the human extremities. So as we can see, the model can produce estimates of muscle force and joint moments using joint angles and EMG data. But before we can run the model, though, the model has to be calibrated. And calibration is necessary to determine a number of parameters that characterize how muscle activates and how muscle contracts. Now, these parameters vary non-linearly across subjects, and they are very difficult to be derived from the literature or experimentally. So the idea here is to use an optimization approach. And the idea is to actually start off this optimization process giving the model a generic set of parameters that describe an average subject, and the model will use these parameters together with input data taken from a pool of calibration trials, and will produce estimates of muscle force and joint moments. And in this step, we now have a third input, which are the experimental joint moments. And the idea is to continuously adjust the model's parameters until we can minimize the mismatch between predicted and experimental joint moments. And we do this over a variety of motor tasks, including forward walking, running, crossover, and sidestepping. So at the end of this step, we have an optimal set of parameters that allow the model to run in open loop. So now the model is able to produce dynamically consistent estimates of muscle force and joint moments univocally as a function of joint angles and muscle excitation. And during the validation step, we ran the model using novel motor trials that were not used during the calibration, and this trial reflected different way of contraction strategies. And the idea was to see if the model was robust enough to be able to predict dynamically consistent joint moments about multiple degrees of freedom during novel trials, and to account for different ways that muscle activate. And this would make the model a fully predictive system. So here are some experimental results that show how the calibrated model running in open loop was able to predict the joint moments produced about 6 degrees of freedom in the lower extremity. So this included hip flexion extension, hip adduction abduction, hip internal external rotation, knee flexion extension, ankle plantar dorsiflexion, and ankle subtalar flexion. And this was done during crossover, sidestepping, forward walking, and running. So as we can see, the model was able to use EMG data and predict estimates of joint moments that matched the reference ones. And the interesting point here is that the calibrated model is flexible, and to a certain extent is also generic, because it can be operated on a variety of tasks that are biomechanically different with each other and underlay different muscle contraction strategies. So this flexibility in the calibrated model is quite important in the context of neurorehabilitation technologies. And with the next slide, I'd like to talk a little bit about this. So the key point is that the EMG-dependent predictions of muscle force and joint moments can actually be used to implement proportional control in exoskeletons. And in this example here, we can see how a very simple EMG-driven model of the knee joint was able to use EMG signals taken from 3 extensor and 3 flexor knee muscles and convert the experimental images into estimates of muscle force and joint flexion extension moment. And we can see here on this graph, the green curve represents the flexion extension moment predicted from the EMG signal. So this is actually the strength in the subject's muscle. And the blue curve, I hope you can see it, is the additional torque that the exoskeleton is supplying to the subject, and that's determined as functions of the torque predicted from the subject. So the interesting point here is that EMG-driven modeling allows understanding the strength of an individual, and this is important to set the exoskeleton support in order to provide additional torque as much as it's needed. So this allows keeping the subject active throughout the rehabilitation treatment and fully engaged. So using EMG data as an input for our model, we can predict output variables that can be useful and it can be effective in the implementations of control system in exoskeletons. In this context, we would need up to 16 electrodes to cover the major muscles in the lower extremity and to cover the major degrees of freedom. Now, having 16 sensors could be limitations in the context of real applications, daily life applications. With the next slide, I'd like to talk about another methodologies that we developed, and this methodologies actually allows removing this high number of sensors. And with these methodologies, we're actually driving the model. Instead of using experimental estimates of the EMG data, we are driving the model using a set of Gaussian curves, which are completely parameterized as functions of the gauge cycle. And the idea is to use this set of Gaussian curves to define very simple strategies of muscle recruitment that we can use to simulate muscle activations throughout locomotion. And I will explain later, these muscle strategies are actually reflections of the central pattern generating networks, which are believed to be encoded in the spinal cord. So going back to the scheme that summarizes the structure of this presentation, we now are working at this level. So we're going to use muscle synergies reflecting the spinal circuitries as an input to the model. So muscle synergies allow reducing the dimensionality of EMG signals. And the basic idea is that the same modularity that exists in the spinal cord can also be observed at the muscular level too. So this means that the recruitment of a large number of muscles can be explained by a small number of excitation primitives. And the key point is that this modularity can also be observed directly from EMG signals. So in this scenario, the EMG signals that we are able to record using surface electromyography are the result of the activations of individual muscles filtered by the skin tissues and affected by crosstalk. So according to the muscle synergy theory, the activations of individual muscles are not independent with each other, but they are actually explained by the activations of a smaller set of excitation primitives, which are combined together according to some rules called motor modules. So the linear combinations of this small number of excitation primitives allows reconstructing the large repertoire of activations that we can observe from EMG signals. So in this, according to this idea, in this study, we're not using EMG signals anymore as an input for the model, but we're using these excitation primitives. Now, due to time constraints, I'm not going to explain how we can derive these primitives from EMG data, But I'll be more than happy to further discuss this in the question session if any of you is interested. So we now have this low-dimensional set of excitation primitives, and this determines an initial recruitment of muscle groups in our model. And using the terminology of Ivanenko and colleagues, this set of Gaussian curves can be seen as an impulsive controller because the onset of one excitation primitive is basically determining the recruitment of a group of synergistic muscles. So as the gauge cycle progresses, these excitation primitives will spike and will generate an impulsive and sequential recruitment of groups of muscles. So the idea here is to further refine this initial recruitment to better fit the muscle contraction strategies observed across a variety of tasks, of motor tasks. And we do this by processing these initial excitations using a second-order recursive filter that we use to simulate the muscle twitch in response to the initial excitation. And then we use a nonlinear transfer function to account for the nonlinearity between excitations and muscle force. And the idea here is similar to what I previously explained. Here we adjust a number of muscle-specific parameters until we can minimize the joint moment prediction error across our motor tasks. And at the end of this process, we have a finer mapping between the 5 excitation primitives and the 34 muscle tendon units in the model. And I like to stress the fact that this mapping, which was derived from this calibration, basically represents a best fit of all the recruitment strategies that were observed across these 4 motor tasks. And that's because the calibration process generated a single set of parameters that describes, on average, the dynamics of the 4 tasks. So to better explain this, I will show the example of the peroneus muscle. So here we can see the green curve is the muscle activation predicted from the impulsive controller, and we can see that the activations of the muscle is the same across the 4 motor tasks. So that's always the same, and we can see that it only fits on average the activations produced by EMGs. Our results show that although this impulsive controller is very simple, and it's static in the sense that it's always the same across these 4 motor tasks, when we combine this impulsive controller with accurate estimates of muscle kinematics, then we are able to produce dynamically consistent estimates of joint moments with comparable accuracy to using EMG signals as an input. And here are some experimental results showing this. And as we can see here, the model driven by the excitation primitives was able to predict joint moments about 6 degrees of freedoms during crossover, sidestepping, forward walking, and running. And we can see that the prediction accuracy was comparable to that achieved by using EMGs as an input. The interesting point here is that if we focus on 1 degree of freedom and we compare it across tasks, we can see that the joint function predicted by the model was quite different across the 4 motor tasks. And this was predicted while having the same impulsive controller as an input across the 4 motor tasks. And this was achieved not just during 1 degree of freedom, but across all the degrees of freedom. So this suggests that dynamically different motor tasks may actually share neuromuscular control strategies which are similar and are impulsive in nature. So up to now, I talked how we can use estimates that indirectly reflect the neural drive. So we used EMG signals and muscle synergies, which are an indirect representations of the neural drive to muscles. And with the next few slides, I'd like to talk about how we can use information on motor units as a feed-forward term, as a feed-forward input drive for our model. And that's interesting because motor units are directly related to the activity of motor neurons in the muscles. So the idea here is to use bidimensional electrodes and to record the activity of the muscle. So this is called high-density electromyography. And this allows us to record the activity of the muscle from different locations simultaneously. So this gives us an ensemble of signals. And a good analogy here is that of having a room full of people talking simultaneously, and classical electromyography, which is based on using a single pair of electrodes located in a specific area of the muscle, would be equivalent to have a single microphone in the room, to, in a specific point of the room, to record the overall ensemble of voices of people talking simultaneously. Whereas high-density electromyography, it's like having multiple microphones throughout the room to record voices from different points simultaneously. And the idea is that once we have this accurate recording of the ensemble of signals, we can then decompose it, and we can determine the underlying sources that generated this ensemble. This is a little bit like saying, like trying to differentiate the voices of each individual person from the recording of the people talking simultaneously. And of course, in the terms of muscles, each person represents a motor unit. So applying the composition, we can extract a number of motor units. In this graph here, we can see we were able to extract 50 motor units. So every row represents a motor unit, and every column represents the temporal event at which that specific motor unit was activated. So this gives us a spike train for each motor unit, which basically reflect the activations of pools of motor neurons in the spinal cord, which innervate multiple motor units in a specific muscles. So by combining together these spike trains for all the motor units that we were able to decompose, we can extract the cumulative spike train for a specific muscle. And we can do this for many muscles crossing a joint. We actually did it for the 5 major muscles crossing the joint angle. And we can then combine, we can then use these cumulative spike trains to drive musculoskeletal models, and we can predict the functions at the joint. And our preliminary results show that it's possible to use cumulative spike trains in isometric conditions, and this is quite exciting and it's quite promising. So up to now, I talked how we can use estimates of muscle excitations to drive, to predict the functions in the musculoskeletal system. This is regarded as a forward dynamic model. In this scenario, we can use inverse models which reconstruct all the musculoskeletal transformations in reverse. So starting from the final movement, we can reconstruct in reverse all the musculoskeletal transformations and determine the muscle controls that generated it and the neural excitations responsible for the generations of the final movement. And we did this step using static optimizations, and we were able to reconstruct in reverse all the activations until the generations of synthetic EMG signals. And the idea here was to combine these two models together. So we're combining the forward dynamic model with an inverse dynamic model together, and we're closing the loop. And we now have what I call a hybrid model. And this hybrid model allows transitioning from the configuration in which muscles can be fully driven by experimental EMG signals in a forward dynamic way to the configurations in which the muscle activity can be fully optimized and derived in an inverse dynamic way. But this also allows for all the gradations and shades in between. And this hybrid modeling approach allows addressing some limitations in surface electromyography. a major limitations is the inability of surface electromyography of accessing deeply located muscles. And another limitation is the inability of properly accounting for crosstalk in muscles and also properly representing the actual bandwidth in the neural excitation. So depending on how much we trust our experimental EMGs to begin with, we can decide to adjust them more or less by moving on this line. And we do this process of adjustment for the purpose of improving the estimated joint moment and for the purpose of addressing limitations in surface electromyography. And the point here is to find the minimum level of adjustment that gives us the best improve in joint moment prediction accuracy. And here we can see in this graph how we were able to minimally adjust linear envelopes for all the muscles in our model. So the red curves here represent the experimental EMGs, and the gray curves are the linear envelopes which were minimally adjusted. And as we can see that for many muscles, the adjusted EMGs are very close to the experimental one. And for some muscles, we had a substantial level of adjustment. And on top of that, we were able to predict, to estimate the activity of deeply located muscles like the psoas and the iliacus, for which we didn't have experimental EMGs to begin with. And the idea is that using this minimally adjusted linear envelope, we can improve much better the predictions of joint moments about 6 degrees of freedom. And the ability of predicting dynamically consistent estimates of all the muscle crossing the hip is really important to study the dynamics of the hip joint. So in this case, we can have a very nice estimate of the forces produced by all the muscles crossing the joint, and we can combine these force estimates with finite element modeling and understand how muscles interact with bone and contributes to the remodeling of the bone, and this is really important in the fitting also of internal implants, but also for the prediction of the development of neuromuscular disease. So with this slide, I'm going to start the last part of the presentations, and I'm going to spend a few slides to introduce the demo, which will explain how our model can currently talk with OpenSim. First, I'd like to stress the fact that all the methodologies that I presented in this talk are currently being integrated into a toolbox that has this very long name. So, calibrated, EMG-informed, neuromusculoskeletal modeling toolbox. And as I showed in the presentation, the toolbox is able to use estimates of the neural excitations reflecting different level of reflecting the neural drive at different levels in the neuromuscular pathway. And the toolbox allows choosing different operation modes for the underlying model. So we can decide how we want to implement the mapping between excitation to activation depending on what estimates of muscle excitation we have as an input. We can use different ways to estimate the muscle kinematics from joint angles. We have the possibility of using different tendon models. And we can also decide how many degrees of freedom we and how many muscles we want to run our simulation with. We can then decide to operate the model as a forward dynamic model, so fully driven by excitations, or as an inverse dynamic model, so we can use static optimization, or we can run the model as an hybrid. We can then determine and decide what parameters we want to calibrate in the model and what optimizer we want to use. And we finally can decide if we want to execute the calibrated model offline or in real time for applications of control of assistive devices. And all these operation modes can be dealt by XML, a little bit like it's done in OpenSim at the moment. So as I said before, the model can currently be used within OpenSim. And in the next few slides, Claudio will explain how we were able to achieve this level of interfacing between our toolbox and OpenSim. So I now would like to pass the ball over to you, Claudio.
Thank you, Massimo. Hello, everyone. I hope my microphone works fine. So, as Massimo explained, CineMass is a powerful tool for the simulation of muscle dynamics. As we can see in this slide, it is possible to choose different options and flavors to run CineMass. However, for the integration of CineMass in OpenSim, we selected a specific combination of these options, like the use of EMGs as input, the elastic implementation of the tendon dynamic, and the use of multiple degrees of freedom. Finally, we selected the forward dynamics mode, which means that CineMass will work in open loop full predictive mode. What we want to achieve is an exchange of information between OpenSim and CineMass. OpenSim computes muscle tendon length and moment arms, which are used with experimentally recorded EMGs as input for CineMass. Consequently, CineMass computes the muscle dynamic according to the options previously defined and returns to OpenSim the muscle activations and muscle forces data. The goal is to allow the user to perform EMG-informed simulation within the OpenSim GUI and receive visual feedback of the results. To achieve a complete integration of CineMass in OpenSim, we firstly wrapped CineMass with the OpenSim APIs. Starting from a motion file with angle data, the APIs perform the muscle analysis and feed CineMass with muscle tendon length and moment arms. Secondly, we created a personalized front end to allow the user to select the additional input required by CineMass as experimental EMGs. Finally, we used the new OpenSim scripting feature introduced with the 3.0 version to create a communication layer between the OpenSim GUI and the other software components. Therefore, the user is able to easily interact with CineMass and has access to all the results directly from the OpenSim GUI. Now, I'm going to show the results of this integration with a brief live demonstration. So here is OpenSim, and first of all, we have to open an OpenSim model. This model, as you know, defines the geometry of the muscles and consequently will influence the results of the simulation. Every valid OpenSim model can be used, of course. Second, on the script drop-down menu, we select our script, run CineMass, and so our custom window appears. In this window, we are able to see which is the currently active model in OpenSim, and now we can select the OpenSim motion file that contains the angle, all the angle of our motion. Secondly, we select the EMG file that contains the experimental EMG data relative to the selected trial. And last, we choose the XML file that contains the subject-specific muscle parameters, which have been estimated during the calibration process that Massimo described before. The click on the Run button starts the EMG-informed simulation. So in this moment, the muscle dynamic is being estimated by the APIs and by CineMass. Here it is. The simulation has just finished, and we can see the results of the activation of the muscle. So we can clearly see which muscles are being activated, and this is from CineMass, from our simulation. So Furthermore, it is possible to view the result of this computation directly from the OpenSim standard plot tool. Yeah, so it's possible to plot activations or forces. In this case, activations. Here it is. These are the activation that we computed just before, and yeah, we can see the line moving on the plot. So yes, this is. Oh, and it's possible to plot the forces also. The forces as the results of our simulations of the vastus. Here it is. So this finishes the part with OpenSim. Let's go back to the slide. And so as we have seen, this integration between CineMS and OpenSim makes available EMG-informed simulation directly from the OpenSim GUI. And before letting Massimo to conclude the presentation, I would like to thank Monica Reggiani. She's one of the main developers of CineMS, along with Massimo and I. And thanks to her, we have such a modular and powerful software at CineMS. So now I'll let Massimo to finish and to give a brief summary of what we have seen today.
All right, so thanks, Claudio, for a nice demonstration. I actually forgot to say that our toolbox will be made available on SimTK, and to begin with, it will be made available as in the form of executable. So with this slide, I'd like to summarize what has been said so far. So our approach is based on collecting experimental data reflecting directly or indirectly the neural drive to muscles, and uses estimates of muscle excitations within our model to be able to predict the resulting functions in the musculoskeletal system. This gives us the possibility of using muscle excitations to understand how muscles interact with bones, but also to implement proportional control in assistive devices, including exoskeletons or prostheses. We can then exploit the modularity in muscles to simplify the complexity of EMG data, and this allows us to implement proportional control in assistive devices even when EMG data may not be entirely available from the patient's muscles. Finally, we can use high-density electromyography to gain an insight into how individual motor units activate within a muscle, and this is important to better understand how the neurophysiologic world is connected to the musculoskeletal world. This can also have important implications for the development of myoelectric control in prosthesis and power orthosis. So with this slide, I'd like to conclude our presentations, and I'd like to thank you all for your attention.
All right, thank you, Massimo and Claudio, for a great talk. We can now go ahead and get started with the Q&A session. So here's a little diagram to show you how to ask questions. So you'll use the Q&A panel, so all the questions will be text-based. You want to find the Q&A panel in your WebEx controls, and then type in your question and make sure you choose to ask all panelists. So with that, we'll go ahead and get started. We already have a couple questions that have come in. The first is the practical one. It's from Ching Sai, and his question is, how much time does it take for the model calibration process to complete? And I'll actually add on a second question related to that. So how long does it take? As well, do you know if you have you calibrate a model to a particular person, if they come back a week later, a month later, do you have to recalibrate? I assume you do, since the EMG placement might have moved, but you guys can comment on that.
All right. So thanks for these are actually great questions. First of all, the calibration process is quite quick, and that's also thanks to Claudio and Monica Reggiani, who really optimized the code. So it really depends on how we operate the model. So the calibration time varies as a function of the tandem model used. If we use a stiff tandem model, the calibration on one subject using 6 to 8 trials for calibration, it runs in 2 to 3 minutes. And using an elastic tendon, it takes longer. And I actually let Claudio tell how much it would take with an elastic tendon.
Well, doesn't take so much longer. It's about 12, 15 minutes maximum. So with the same setting as you described. So
right.
Yes.
Right. And so calibration is quite fast, and the execution of the model is the model can be run in real time. So the model runs within the muscle electromechanical delay. So we can have an estimate of muscle force and joint moments every 20 milliseconds. And with regards to the addition of Gen, it has been proved that the parameters that are calibrated in the model can be divided into two groups. There are a number of parameters that determine the EMG to activation transfer function, and there are a number of parameters that determine the morphology of the muscles. So these include tendon slack length and optimal fiber length. And whereas the muscles related to the EMG to activation transformations are, we have two filtering coefficients which determine how the internal filter of the model respond to an impulsive input. And we then have a parameter which determines the nonlinearity between excitations and force. And so what it was proved by the work of David Lloyd and Tom Buchanan and Tor Bezier and colleagues is that it is possible to calibrate once for all the anatomical parameters, meaning tendon slack length and optimal fiber length, because those are related to the geometry, to the anatomy of a subject. And in an adult subject, these parameters don't change. So these parameters need to be calibrated once for all. On the other hand, the parameters that are associated to the EMG to activation transformations, they are quite sensitive to the placements of the EMGs and also to a number of physiological conditions like blood pressure, impedance on the skin, and so on. So these ones have to be recalibrated for every testing session.
Okay, great. Thanks. That's a very clear answer to those questions. So we have a couple more questions related to calibration. Massimo, can I remind you to really speak up and speak into the microphone, just because there's a little bit of a difference between your volume and Claudio's volume. So we don't want to blow people's ears out. Okay, so the next question is from Stefan Lambrecht. His question is, what is the influence of the various calibration tasks that you use, say running, sidestepping, crossover, on the predictive value? In other words, how does the accuracy improve by including more than one task in the calibration?
Right. So basically, the idea of adding tasks which are biomechanically different with each other, it's not. So we don't do this because for the purpose of improving the accuracy of the calibration. We do this for the purpose of accounting for a larger spectrum of muscle recruitment strategies. The calibration would work the best if we would use just one trial for calibration. So this would give us a perfect fit. This would give us a parameter set that would fit that specific trial perfectly. But then what would happen is that when we run the model in open loop, the model would not be predictive, because the model would be fitted completely to just one specific trial. So in order to improve the predictive ability of the model, we include a larger variety of tasks that allows us to represent the full spectrum of muscle activations. But this, in principle, decreases the overall. This increases the RMS error between predicted and experimental EMGs, but on the other hand, it also increases the predictive ability of the model. So we can run the model on many novel trials, and the model will stay dynamically consistent.
Okay, great. So is the calibration process, is that going to be part of the toolbox that you're going to share on SimTK?
Yes, yes.
Okay.
The idea is to also provide the calibration as a toolbox. Probably Claudio can say a few more words to say what we are planning to release in the future.
Yes, so the idea is to release this toolbox, even though we want to be sure that everything runs smooth for the user and there are no usual problems or anything like that. But we are planning to release the calibration as well, because otherwise the model can't work properly without a calibration process. I'm going to implement the calibration in a similar way as we have done with the execution part of the model itself. So what we have seen, there will be something similar and easy to use as the execution of CNMS as the estimation of the muscle forces. So it will be a similar thing, still with the scripting. And yes, it will be available in the, I think we need some months still to be sure that everything is perfect. So that's our plan.
Great. That answered another question that had come in when you were planning the release.
If I can add, so what we want to do is, before we release the software, we want to make sure that the software is bug-free. So this is gonna take. For this, we're gonna need a couple of extra weeks, probably a few months. But at the end, we will have a first release of the toolbox on SimTK, and this will feature an EMG-driven model running in open loop with the possibility of calibration.
Great, that sounds great. Okay, so let's move on to another question. Let's see, where did it go? Okay, so this is a question from Seamus Chamanadu. Apologies if I mispronounced. It says, great talk, thank you. Can you comment on considering mechanical behavior of the muscle introduced in the CINMS toolbox? In other words, to predict the forces through an electromechanical model using a constitutive model plus the activation inputs that you referred to.
I'm not sure if I understood the question. So the question is related to how the process of excitations of muscle and force production works.
Yeah, I think the question is whether you've considered using a constitutive model of that process.
Right. So what we do at this time So we have a blog that models the transformation between excitations and activations, and as I explained, this is important to account for the nonlinearities in muscles, which actually may vary across muscles. And then, of course, our muscle model is a hill type muscle model. So it's a standard hill type muscle model, which actually was modified by the many people who contributed, and I actually acknowledge them at the beginning of my talk. So this features a nice elastic tendon dynamics, a nice way to predict the optimal fiber length and how this changes into the functions of muscle excitations. But for the moment, we're planning to keep this model as main muscle implementations in our toolbox. But of course, we're open to extend the model to different methodologies and approaches in the future, for sure.
Okay. Now we have a couple questions about the parameters of your model. First, from Walid Hassani, he's wondering how you know that the results you get, say, for muscle optimal length are a good value, since you're just optimizing to fitted data. And related to that, Jonathan Slowick asks, how much do the parameters typically vary when you use different calibration runs for different people?
Yes, so these are also very nice questions, so thanks for these questions. So the key point of the calibration is that it is a constrained optimization process. This means that parameters are allowed to vary within physiological boundaries. So we don't allow parameters to vary indefinitely. So, for example, tendon slack length and optimal fiber length, we can only adjust them within a 2.5% of the initial estimates. And this initial estimate of tendon slack length and optimal fiber length is derived using a methodology which was published by Windy and colleagues on Journal of Biomechanics, if I remember, in 2008. So this is basically a little bit like the linear scaling in OpenSim. In this case, we are actually able to scale the non-linearly the optimal fiber length and the tendon slack length to achieve a nice starting point. And then these parameters can be further adjusted in the calibrations, and they only are allowed to vary between a 2.5% of their initial value. And same story for the EMG to activation parameter. So we have two filtering coefficients, and these parameters, they are also constrained. They vary between -1 and 0, and the idea is to basically keep the filter stable. And then we have the nonlinear shape factor, which varies between -3 and 0, and this was published by Buchanan and colleagues, and it is also constrained. What we showed is that there is consistency in the calibrations of the anatomical parameters. Those are very consistent. And whereas the calibration of the EMG to activation parameters is way less consistent. So by using data from different data sets, we would have substantially different results in those parameters.
Okay, great. Thank you for clarifying that. I think we are about out of time, so I'm going to go ahead and close the Q&A session. Thanks for all the great questions and your clear answers, Massimo and Claudio. Can you go ahead and move me to the next slide? The next one.
Oh, sorry.
Yeah. Okay, so we definitely want to acknowledge the funding sources for the OpenSim project. OpenSim in this webinar series are supported by several grants from the NIH and the EU, including an NIH grant that funds our National Center for Simulation and Rehabilitation Research. We're also supported by the DARPA Warrior Web effort. I want to again thank you all for a great discussion. You can find information about the center, upcoming events, future webinars, and other resources from the OpenSim community on our website. And then also please fill out the survey that will appear in a pop-up window at the conclusion of the webinar. This will help us improve the webinars and choose future upcoming topics. So thank you again for participating in our webinar, and we hope you'll continue to stay involved with OpenSim. Thank you.